Hi, I'm Chase Schubert, and I'm an upcoming junior at Whitman College in Walla Walla, Washington. This summer, I'm an REU intern here at Dauphin Island Sea Lab, and I'm in the Marine Ecotoxicology and Biodiscovery Lab working with Dr. Alison Robertson. I'm evaluating traditional approaches for the detection of ciguatoxin-contaminated fish, specifically to see if ants are able to determine the differences between toxic and non-toxic tissue. If they can, this might indicate a semi-volatile biomarker in the fish that can be used for rapid detection. Ciguatera poisoning is a global seafood-borne illness caused by the algal-derived neurotoxins called ciguatoxins. It is estimated that up to 500,000 people are affected annually. In some endemic areas, as many as 10% of seafood consumers have been affected. These toxins are naturally produced by the benthic microalgae Biodiscovery and are colorless, odorless, and extremely stable as chemical compounds. The source dinoflagellates, pictured on screen under a microscope, are commonly found in coral reef ecosystems, living attached to substrates such as macroalgae and seagrasses. The ciguatoxin poisons produced by Biodiscovery are then distributed throughout marine food webs following consumption by herbivores like invertebrates and fish. They are then bioaccumulated and modified in these species, causing the toxins to be transferred to higher trophic levels. Of particular note, this includes species targeted for human consumption, such as snappers and groupers, both of which are displayed in our very own Dauphin Island aquarium. As a series of neurotoxins, consumption of fish contaminated with ciguatoxins can cause a variety of neurologic and gastrointestinal symptoms. While the prevalence and severity of ciguatera poisoning is a significant concern, to date there are no reliable and sensitive rapid tests for the detection of ciguatoxins in seafood, making prevention difficult. Advanced laboratory methods are available, but these are expensive, time-consuming, and require sophisticated instrumentation. However, island communities across the world have reported traditional strategies for toxin detection that may have some scientific basis. In my study, I'm going to focus on one of these specific folk tests. Our experiment is broken into two parts: 1, field testing, and 2, a lab-based study. For both experiments, fish tissue samples were prepared from Cypherina barracuda, more commonly known as the great barracuda. These samples were originally collected from the U.S. Virgin Islands. The first tissue sample was verified as having no toxins, so it will be used as a control, and the second sample was verified to contain Caribbean ciguatoxin 1. In the field study, I am testing if local ant colonies naturally differentiate in reacting to toxic versus non-toxic fish tissue. You might be asking yourself, why ants in the first place? Well, they have an excellent sense of smell, so we think they are good candidates for this sort of work. This is what I've been dedicating most of my time to so far at Dauphin Island Sea Lab. By placing a neutral platform 10 centimeters away from the ant colony's entrance, we can record the natural reactions of the ants, including how many ants swarm the tissue over a set period of time. I'm also recording a variety of other behavioral metrics, and I'm learning tons about ants. In the second part of the study, I will be intentionally training ants that have been raised in our lab for the past few weeks. Now, that might sound crazy, but in prior studies, ants have been trained to detect differences between cancerous and non-cancerous human cells, so the precedent does exist for their scientific potential. This will be done with the use of our recently constructed ant arena. I will place a small drop of sugar solution in close proximity to pieces of poisoned fish tissue to create a psychological association between the two stimuli. After being conditioned, we predict ants will identify ciguatoxic tissue by walking towards them, even if other clean tissue samples are present. My work in the Robertson lab has been incredibly fulfilling. I've had such an enjoyable time both in the labs and in the field as an REU intern. I am looking forward to reporting back with some exciting findings. This is Chase Schubert signing off.